Okay, so good morning, everyone, and welcome to the session 72 of Beyond Research, where curiosity meets conversation, co-organized by Biopractify and the ACS chapter of IIT Delhi. And today's session, Decoding Proteins and Antibodies Insights into Modern Biopharmaceutical Analysis, will take us into the analytical backbone of modern biologics, from monoclonal antibodies to complex glycoproteins, and how advanced characterization drives safe, effective, and affordable therapeutics. We are honored to have with us Dr. Sanghati Bhattacharya, senior scientist at Stira Pharmaceuticals, New Jersey, with over 7 years of experience across academia and industry. Dr. Bhattacharya specializes in LCMS based analytical methods development, working extensively on monoclonal antibodies, complex biologics, and N-glycans within FDA and ICH regulatory framework. Her work focuses on advancing next generation biotherapeutics, making them more robust and accessible. Beyond her research, she is an active scientific contributor with international publications, invited talks, reviewer roles, and strong engagement in teaching and mentorship. Please join me in warmly welcoming Dr. Sanghati Bhattacharya. Welcome, ma'am, and over to you.
Thank you. Thank you for the wonderful introduction. And how do I share my screen? So can you, okay, just 1 minute.
Yeah, I've stopped sharing. You might be able to now.
Okay. I am not able to guess.
It could be in the top right corner.
Yeah, that I have seen, but it is not coming. Just 1 minute. Somehow it is not coming. My desktop thing, it's not coming. I don't know why.
Do you want me to share? I'll share your slides.
That can be done. Somehow it is not coming to my.
Sure. You can either reply on the same email or I'll text you.
Just 1 minute. I'll try once more time and let's see. Okay, I'll. Yeah, this is in WhatsApp. Okay. Should I share it? Should I share it with you in WhatsApp or email?
Whichever is more convenient for you.
Okay. I don't know why somehow it is not. I'm not getting that sharing my window option.
Are you joining through browser?
Yes.
Okay, if you try maybe Chrome or Mozilla, then
I'm
it
sending.
could.
It's okay. I'm sending
Okay.
you. You can share. It's okay. I have shared. Did you received? I have shared through WhatsApp.
Not yet.
Not yet.
Yeah, got it.
Okay. I don't know why it is not.
I hope it is visible, my screen. Is my screen visible?
Yes, it's visible.
Okay.
Okay. Okay. Should I start now?
Yes, you can start.
Hi, everyone, and I'm really sorry because I have postponed it couple of times. And for everyone, my name is Sanghati, and I am an analytical researcher by training. I was born and brought up in India, therefore most of my studies I have done from there itself. And my specialization is liquid chromatography mass spectrometry. I'm currently working as an analytical researcher, as a senior scientist at Stira Pharma. And today I'm going to talk about insights of modern biopharmaceutical analysis because I'm an analytical researcher by training. And I'm going to talk about how to decode protein and majorly large molecule biologics, majorly monoclonal antibodies, and other biologics by analytical techniques. Next slide. Okay, so these are the content I'm going to cover. So first I'm going to talk about the history or development, how monoclonal antibody developed, and then followed by biotechnology as in technique, and then biosimilars, which are very much into the prominent scenario of today's biological or biotherapeutics aspect. And then the regulatory guidelines, how it evolved from maybe 1980s or before that, and current scenario of it. And then I'm going to talk about the major workflow or protocol or pipeline of novel drug development, how it is getting manufactured from upstream development, followed by downstream processing to purification, and then why it is coming to analytical division, why there is a need to understand what are the critical quality attributes, what are the process related variants, and how we have to assess or identify the structural heterogeneities or structural variations which may be formed during process variability or process related things, and then followed by confirmation or validation before commercialization. Then I'm going to talk about major biotherapeutics of which we are going to deal with, and followed by monoclonal antibody structure, and the structural elucidation using liquid chromatography mass spectrometry, how large molecule biologics in terms of company point of view, how we are going to assess, and then critical quality attributes, and then impurity assessment of majorly large molecule biotherapeutics, and then advanced analytical techniques which has been used from not even a decades, maybe 4 5 years, which are very advanced, and currently not only academia, industry are also working on these things. Next slide. So this is the biological development history, and the orange color dots are representing monoclonal antibody development from early 90s, maybe 1986. First murine antibody got developed, or monoclonal antibody got developed, and followed by 1996, that is humanized monoclonal antibody, followed by 1994, that is chimeric monoclonal antibody. 2002, it's the first human monoclonal antibody. So if I'm talking about the murine monoclonal antibody, humanized monoclonal antibody, chimeric monoclonal antibody, and the first human monoclonal antibody, then it is coming towards, so human monoclonal antibody as the most privileged and predominantly used one for today's scenario. And it is if you are coming from murine to human monoclonal antibody, it is reducing the immunogenicity. So if we are talking about monoclonal antibody, we are always focusing on how we can reduce immunogenicity. After a couple of slides, I'm going to talk about the monoclonal antibody structure and function. Then we can we can understand it in a better way, how we can reduce immunogenicity in that aspect. And the blue color dots represent the advance in biotechnological as a subject, or biotechnology as a technology. So early 1890s, it has been started and followed by recombinant DNA technology, then discovering the insulin and approved drug, biotech drug through FDA, US FDA, and then the cloning of sheep, which we already knew. It was developed 1997. Name was, if I'm not wrong, Dolly or something. And then followed by 2001, and followed by 2003, Human Genome Project got published. So all this, from early 80s to early 1890s to 2001, it has been developed in a good way. And if we are talking about biologics and biosimilar products, it also has been started from 1978. First biological medication was developed is a insulin, and then followed by growth hormone, and then different growth hormone and different kind of biologics are developed through the 1978, 1981, 1989, followed by 2000, 2006, 8, and finally 2012, 14 biosimilar got approved through European Union. So what is biosimilar? So biosimilar are similar but not identical. That means, suppose we are having a drug which is serving similar same function after injecting or after administrating in a human body, but they however, if they are similar in terms of their function, but in the how they are getting manufactured, that is different. That means their content of, or the excipient, or whatever we need to manufacture a large molecule biologics or a drug product, that are different. So if I'm talking about as a simple term, I can talk about like any ice cream, like what to say, butterscotch ice cream from different company, like Amul butterscotch ice cream and the quality butterscotch ice cream. It is giving the exact same taste, not exact, but somehow it will give us the same taste or kind of thing. but the ingredients for making it are different from company to company. So if that is the case for drug product, we are calling it biosimilar. So biosimilar is very much necessary for the country, third world country like India, because we are having we are actually having a high population country, and we don't have that much money to bear costly drug like autoimmune disease drug or cancer kind of thing, drug we are needed for cancer or this kind of disease. So in those kind of scenario, we need biosimilar, because if we are having biosimilar, those are there are very much, we can reduce cost in terms of manufacturing and all these things. Now if we see the green dots, those are guidelines of regulatory terms. So we have two different guidelines in terms of regulatory term, that is European Union, and that regulatory guideline we call it as EMA and US FDA. So you can see from 2001, majorly European Union, they started drafting the guideline for biosimilars, and it got published first time 2005, and then Japan and Canada adopt the EMA guideline, and US FDA approval of biosimilar has been taken place from 2010, followed by 2012, and finally approximately 2017 or 18, the biosimilar market has been accelerated in a good way. Next. Next slide. Okay. This is the pipeline of novel drug development. So, if we are talking about biologics or any kind of drug development, there are multiple ways or path or phase where we have to take a drug from the very first phase to the end phase to make it commercialization. So very first stage is target validation, that is taken longer, approximately 1.5 years. And we are taking a lots and lots of candidates, which we are making screening it out and taking it for the further phase, that is compound screening. And approximately more than 10,000 candidates has been chosen for this phase, and again this phase takes around 1.5 years. And in next phase is the lead optimization, where drug like properties like solubility, permeability, ADME, plasma, PK, efficacy, toxicity, those has been identified or we experimented. And then from the more than 10,000 candidates, we select approximately 250 candidates. So the pool has been become very less. And from them, from that phase, that is lead optimization, we are taking the approximately 250 candidates into next phase, that is preclinical test. Again, it takes around 1 years, and we are selecting approximately 10 to 2 candidates for next phase, that is phase 1. In phase 1, PKPD, dose escalation, toxicity, release testing. That means if we are administering a drug in terms of tablet, in terms of injectable, in terms of capsule, then how it release after injected in human body, how absorption takes place. Those are the tests we are doing in the phase 1, and it approximately takes 1.5 years. And from the phase 1, we are going to the phase 2 and phase 3, approximately then 4 candidates followed by 2 candidates, and finally we approved only 1 candidate. That also takes around after testing dosage, efficacy, and toxicity. And from more than 10,000 candidates, we are narrowed it down to 1 candidate to make it commercialization. So we can understand the whole process is very much time consuming. It approximately takes around 15 to 20 years to develop one novel drug, and also it is cost effective, because all the phase I'm talking about or I have listed over here, it's not only one particular testing we are doing. We are actually doing multiple orthogonal techniques for one particular assessment. That means, suppose we are dealing with toxicity, then to make it finalized, so how many candidates are not toxic for the very that particular phase, we are doing approximately 200 experiment. So you can understand how many experiment and how much money is there to make it commercialization of only one drug molecule. Next slide. Now, if I talk about only biotherapeutics or biotherapeutics protein, then we can categorize it into three major division. If the particular molecule is 100 to 1000 Dalton size range, then we called it as small molecule. If the particular biotherapeutics is 1 to 30 kilodalton, we called it as peptides or biotherapeutic peptides. And if that particular molecule is more than 30 kilodalton, we termed it as biologics. So for the further subsequent slide, I'm going to talk about biologics, majorly in monoclonal antibodies, that is 150 kilodalton in size range, because I am majorly working on monoclonal antibody and large molecule biotherapeutics. So I will going to talk about these and how by analytical tools and techniques we identify, we assess, and we validate the particular product. Next. So this is the, yeah. Okay, so this is the structure of a monoclonal antibody, how it is actually looking like. You can see it is a Y-like structure, and the uppermost portion we called it as Fab region, and the below portion we called it as FC region, and the junction we called it as hinge region. So in the Fab region, okay, sorry. Okay, so in the Fab region, you can see there are two division. One is upper, that is green and yellow. We call it as variable region because this particular region is specific for antigen binding. And the dark blue region are the constant region, or you can say it is a crystallizable region. You can see this black color. I don't have any. I cannot show the pointer. So okay, so black colors stand or chain, that those are the disulfide linkage. So in a monoclonal antibody, we are having 4 inter and 12 intra-chain disulfide linkage. So these black color lines, those are inter-disulfide linkage. Those help actually to make the structures intact. So the the main Y kind of structure is heavy chain, and the side one we called it as light chain. So the uppermost tip of the variable region is very much the way I told, it is very much specific for antigen binding, and there the monoclonal antibody have specific code. They have a specific code which can which can help to bind different kind of antigen, and it is actually during the binding of with any specific antigen, it can change its structure. So the whole structure of the monoclonal antibody is, it never changed, but the variable region can change. So the constant region, that is another reason we call it as constant region, because it cannot change, it cannot move, but variable region can, and it make it structure in a way so that different antigen can come and bind, and it can serve the specific function. Next slide. So this is the more elucidation of the structure of monoclonal antibody, and I have pointed out the major region or major amino acid position which are very much susceptible during the process point of production. It can undergo various different kind of modification, so that when a monoclonal antibody is getting produced from very beginning, from upstream development, and then it comes to downstream processing for purification. In this, during that time, various process related variant can be forms, because upstream develop upstream development, we are producing the monoclonal antibody and we are subsequently purifying the protein. But during purification, it is getting nurtured with various different excipient, various different buffers, various different pH, and all these things. During those time, these are the specific region or hotspot, we can say, those can get modified. So I can list it out some of those, like asparagine, that is very much susceptible for isomerization. Methionine, tryptophan, those are very much susceptible for the oxidation. Then N-terminal pyroglutamate formation, then thioether formation, then glycosylation. Okay, another major thing in monoclonal antibody is it is having two specific region where N-glycan can attach. The pink color Y-shaped thing is a glycan. So each heavy chain having one specific region where one glycan can come and attach. But the glycan attachment or glycan, it is high glycan region of monoclonal antibody is highly heterogeneous. So it is also the N-glycan is very much specific for the specific function of monoclonal antibody. So what kind of glycan is getting attached, it depends what kind of function it will going to serve. So these are the so now if during the process related thing or process during processing of the monoclonal antibody, like purification and then analytical characterization, it can get different post translation modification also can be happen. And yeah, those are the major, I think I have listed out most of them. Okay, next slide. Okay, now after uh, purification, oh, so first it is the upstream development. Once it is done, it is coming to the purification or downstream processing. Once it is done, then it comes to analytical division. So what is the main function of analytical division? They actually try to figure it out or identify the particular monoclonal antibody which we are thinking we are having in a purified form, that is there or not, or how purified it is, how purified form it is in present in that particular sample. So what we do, we usually analyze in a structural point of view. So we three kind of structural analysis we do by using, I am just talking about liquid chromatography, mass spectrometry based structural analysis. So majorly we do top-down proteomics, middle-level analysis, and bottom-up proteomics. So what is top-down proteomics? That is intact mass analysis. That means, suppose we know our monoclonal antibody is approximately 150 kilodalton. So we will select one particular liquid chromatography, that can be reverse phase chromatography, that can be size exclusion chromatography, or anything according to your interest or that person's interest or the manufacturer's interest. And then we will elute out that particular monoclonal antibody if it is through reverse phase analysis, or if it is size based analysis or whatever. And then we connect that liquid chromatography through mass spectrometry so that we can identify the exact mass of that particular molecule. So that is top down analysis, because we are not doing anything, we are directly injecting the sample and we are getting the exact mass. So after injecting, if we are getting exact mass of approximately 150 kilodalton or something, then we can tell, okay, that particular product is belongs to that particular monoclonal antibody we are talking about, or we are thinking about, or we are working with. Next phase of analysis is middle of analysis or middle level analysis. Here what we do, we use some enzymatic treatment to chop down the monoclonal antibody. So there various kind of enzyme can be used. So one type of analysis, I mean enzyme, it chopped the FC region from the Fab region. Another type of reduction you can do using DTTC tape, that is, it reduce the um, heavy chain from the light chain. There, I mean, there are various different reducing agent and proteolytic enzyme to chop down the subunit, the Fab region, the separation of LC with HC, this kind of thing. And then we elute out again using a, I mean, any kind of chromatographic technique, like it could be reverse fed chromatography, it could be size exclusion chromatography, and then we can couple it with mass spectrometry so that we can get the mass. But in that case, we will going to get mass of the reduced protein, and the what kind of reduction we have done, that we know. So accordingly, we can get the reduction. So I will, next slide, I will going to talk more about this. And bottom up proteomics is we will completely chopped our protein, that is, we are making fragments or peptides out of it, and then that is also using an enzymatic treatment. And that is actually we have, I mean, the peptide mapping is a very large sample preparation thing. And once it is done, then we have to I mean, it's kind of puzzle up. So once we are getting the peptides, we have to puzzle, like in childhood, the way we solve the puzzle, we have to solve the puzzle of peptide, making the one intact protein. So that is we call protein bottom up proteomics. So if we can solve the puzzle, that means whatever we have done or whatever protein we are working with, that is same. So that is a confirmatory test. Next slide. Okay, so how it is actually look like. So if we are working with top-down proteomics, that is intact level analysis, so these are the data you can get. So visually how it looks when it come out from the detector of the mass spectrometry. So you can see 23 plus and 6445.4. That means that is the charge distribution or charge envelope which has been generated through mass spectrometry. And if you multiply this with, so that is mass by m by z, and the charge is the 23 plus. So if you multiply, you will be getting the exact mass of the particular monoclonal antibody. And the right panel, it is showing the different glyco variants of that particular monoclonal antibody, how it looks when you look through mass spectrometric view. Next. So this is the middle level proteomics, middle level analysis. So first, I mean, uppermost position region of the slide, it is showing the PNGS or IDS or DTT. So that is different enzymatic treatment. So if you are dealing with PNGS, it will reduce or it will chopped off the glycan portion only. So you are having a monoclonal antibody without glycan. Now you are treating with IDS or DTT, that is reduction. So IDS will work differently, DTT will work differently. So IDS will reduce, it is I mean subdivision analysis, and DTT, it will reduce the light chain and heavy chain. Now if you are running it through liquid chromatography mass spectrometry, that means in liquid chromatography you have to select your particular column of your interest, like it can be reverse phase, it can be anything, and then followed by mass spectrometry, that is for detection. And below you can see, so FC2, that means we have two FC region, and LC, that is we have only one LC region and FT region. So you will be getting three different picks for FC, for LC and FT. So these are the, middle level analysis. If you are below panel, it is showing the reduction only. So if you are if you are only reducing your protein using DTT or CTAP or any kind of this kind of proteolytic enzyme, your protein will reduced into light chain and heavy chain. And if you run through liquid chromatography, you will be getting two peaks. One peak will be heavy chain, one peak will determine the light chain. And if you couple it with the mass spectrometry, it will give you the exact mass of heavy chain and light chain, and that will be a confirmatory for your protein. Next slide. Okay, so this is the yeah. So this is the bottom-up proteomics, and I tried to make it very simple. The conventional method of bottom-up proteomics. And you have to, you suppose you are having native proteins, so you have to denature your protein, and then you have to so there are denaturing agent like urea. Some people use SDS, and then you have to reduce your protein, then alkylation you have to do. So reduction, after reduction, and there are different agent like DTT and then IAM and many other reagent also. And once your protein are reduced and alkylated, then you are introducing enzyme, you are doing the enzymatic digestion. So most common used enzymes are trypsin, chymotrypsin, lucy. So those are all approximately having specific region to cut in your protein. So once you are once you are treated your protein with a specific enzyme of your interest, you know where should be the cut, and accordingly, after LCMS analysis, you will be getting the sequence coverage. So left side panel, how liquid chromatography mass spectrometry data looks like after peptide mapping, and below that multiple color thing, that is the sequence variant analysis. That means how much, after puzzling up, now if you are having enzymatic digested peptides, that means you have a puzzle to solve. So the colorful panel, it is showing what are the puzzle they are getting solved, and what are the region where they didn't have any particular peptide to get resolved. So once you get that data, then you can identify how much your peptide got. It is what is the score of your puzzle. So if it is more than 90%, then you can confirm, not 90%, most of the cases, if you are doing with the purest protein or drug product, mostly you are getting more than 95% of the sequence coverage, and that itself tells that your protein is the one which you are looking for. Next. Okay, so there are another term which we use majorly, that is critical quality attributes of protein. That is very necessary, because once manufacture once production of a monoclonal antibody has been done, then we have to before even purification or after purification, we have to understand what are the critical quality attributes it may generate during the process related variants, during the process. So for that, why it is necessary? Because we have to minimize the immunogenicity, and the way we are assessing the critical quality attributes, the term we use the quality by design. Because if we there are multiple ways. You can go with traditional ways, you can go with advanced way. I have listed out some of the traditional way and the followed by advanced way, how people nowadays, people are using, industry using to analyze critical quality attributes of large molecule biologics. Next. Okay, so this is the size variant analysis. So suppose just now I talked about critical quality attributes. There are multiple critical quality attributes, and the one main critical quality attributes for large molecule biologics are is size variants. That means, suppose we have a manufactured monoclonal antibody that we called it as drug substance. And after purification, we don't know what are the variants it may generated. So during the process, maybe it got dimerization, it got trimerization, it can make multimer, it can get fragmented. So how do we understand what are the variants it may generate? It may generate it, and what are the what is a number in terms of quantification. So I mean, is that within the range to make it safe commercialization or not? So the main IgG, this is the chromatogram of size exclusion chromatography. So in as I'm talking about liquid chromatography mass spectrometry, so I'm showing the size variant analysis through size exclusion chromatography. There are multiple other techniques also to understand size variant, but if you are choosing size exclusion chromatography, then you have to run isocratic elution. That means you have to, with the specific particular column, you have to run your sample, and the 150 IgG monomer is the monomeric peak. That means that is the intact protein, and left side is the product that has been eluted earlier. That means if it is eluted earlier, we call it as high molecular weight species. That means those are higher molecular weight species than our monomer. So it could be dimer, it could be trimer, it could be multimer, it could be anything. And the peaks that those are eluted later on, we called it as low molecular weight species. So it can never be an intact protein. It is always fragmentation of maybe high, heavy chain. It could be light chain. It could be heteromer of heavy chain, light chain. It could be anything. So to know this, we have to again we have to combine this chromatography, that means size exclusion chromatography, with mass spectrometry, so that you can get the exact mass, and we can confirm, like here you can see in each high molecular weight species, it is showing more than 300 kilodalton. That means those are aggregate that has been formed during the manufacturing or during the process, not manufacturing only. And low molecular weight species 1 and 2, you can see those are approximately 100 kilodalton, followed by low molecular weight species 3 and 4. Those are approximately 50 kilodalton in size. Those are not intact, those are the fragment that has been generated during the process. Next. So this is again another kind of size critical quality attribute analysis. This is to understand the acidic variants and basic variant that has been formed during the process. So normally to understand the acidic or basic variant, that is a gradient illusion. You have to choose a specific column, that is maybe a weak cation exchange chromatography or strong cation exchange chromatography. Normally for monoclonal antibody, we generally use weak cation exchange chromatography, and the gradient here you have to implement. And gradient can be two kinds of two one could be gradient of pH, one could be gradient of salt base. So you can use salt base buffer, a salt gradient you can choose, or you can choose a different pH, like for elution, for binding, and you can elute it out through your chromatogram. So here you can see the early eluted peaks are terms at acidic variants, because once you compiled it mass spectrometry, the last panel showing, it is showing like the middle one, it is showing 148058.81. Just before that, it is 147852.31. That means 1 Dalton shift, 1 kilo Dalton shift. That means, as if I mean you I mean if deamidation happens, then Dalton shift has been occurred. So after getting confirmed through mass spectrometry, we can say, okay, yes, it is the deamidated version of the particular main peak. So that is the acidic variant of that particular protein. Followed by, if you consider the basic variant and the purple color panel, you can see the oxidation occur two times, that is single oxidation and double oxidation of the main peak. So in that way, you can understand the process related variant which may be generated in terms of acidic variant, basic variant. We also called it as impurity during the during the process of the manufacturing. Next slide. Okay, so I have listed out some of the analytical techniques and attributes we majorly use. So, that is not we are not limiting over there. I mean, there are multiple, actually multiple, like 200 different techniques for one particular assessment. Like I have listed out here for primary structure analysis, we are using intact mass analysis, reduced mass analysis, peptide mapping analysis. Those are through liquid chromatography, mass spectrometry. But there are several other techniques and other instrumentation also which use to understand primary structure. So all the orthogonal analysis will come up in a solution that the primary structure which we are depicted, that is coming from the same protein. Next. 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 Okay. No, next, before. Next, that's it. So this is, no. Okay, again, one more. Bus, that's it. Yeah. No. Oh my God. Okay, yeah. So this is I just listed out some of the traditional technique before. So this is the most advanced technique. Now it is, maybe couple of years we are using. I mean, globally it has been used maybe not more than 5 years. So this is the technique which we called it as multi-attribute monitoring method, and what we are doing, we are combining different liquid chromatography in a single or double or multi-dimensional chromatographic tech. In a single or two-dimensional platform, and then we coupled it with the mass spectrometry. And the idea is we will going to assess multiple critical quality attributes in a single go. So here you can see we have coupled protein A chromatography with size exclusion chromatography in the first division, and then we are using weak cation exchange chromatography in the next division, and followed by mass spectrometry, so that we can get the glycation of that particular uh, charge variant of the selected size variant for that particular protein. So that means here we are able to identify 4 different critical quality attributes in a single go. I'm not going to talk more about this. And next, I'll just briefly telling the advanced technique people are using nowadays to understand critical quality attributes. Next. Okay, so this is another type of analysis. It's, very much advanced. That is glycan engineering. That means suppose we are having a glycan, and glycan, it is not, it is like post-translational modification, and you cannot have control over producing glycans or a kind of glycan it would produce during manufacturing of the during production of the monoclonal antibody. So actually you have control over it, but it's very difficult, because you have to choose a particular selective cell line, like CHO cell line if we are using, then we know what kind of glycation will going to occur. So another type is CO2 optimization during the production. So that is also a challenging thing. So what is not simplest but way too easy to go kind of method is glycan engineering. So what we are doing over here, we using different kind of enzyme to chop up the glycan, and again we are reattaching of glycan of our interest. Now what will happen? So now, suppose we are reattaching of our particular glycan of our interest. So the monoclonal antibody will not be having heterogeneous glycan. It will be having homogeneous glycan. So what kind of purpose it will solve, or what kind of function it will going to solve, that will be very much significant for disease to disease. So in that way, we can modulate the immunogenicity. So that's a very advanced technique people are using. Next. This is another technique, that is formulation, exchange. So I like suppose we are having different formulation available in the market, then we are, not we, industrially people, what they do, they use different formulation for particular one product or API. API, that means active pharmaceutical ingredients. That is a, but we call it as, like in monoclonal antibody, when it is being produced, we call it as API. So then we analyze analytically with different techniques, and we see what formulation is actually best for which kind of protein or monoclonal antibody. So this is another technique people are using nowadays. Next. I think I'm done. Okay, so these are the people I have worked with, my mentor, my supervisor, my colleagues, and these are the universities I have worked on and the funding agency. And I would like to thank you. And if you have any question, please ask.